Hello and welcome to my protein-binding DNA microarray analysis presentation. So I analyzed the Grainyead-like protein 2 homolog, which is a pivotal transcription factor intricately involved in numerous biological processes, particularly primary neurulation and epithelial development. Acting as a transcription factor, GRHL2 regulates gene expression by directly binding to DNA sequences, thereby influencing crucial gene regulatory networks essential for cell fate, determination, and tissue morphology. The consensus binding sequence for GRHL2 is identified as from 5' to 3' AACCGGTT. So this represents a critical site for its interaction with DNA and where it exerts significant effects on biological processes. GRHL2 plays a crucial role in various developmental and physiological pathways, including primary neurulation and epithelial integrity. In neurulation, GRHL2 is indispensable for proper neural tube closure, and disruptions in its expression are linked to neural tube defects, underscoring its importance in neurodevelopment. Additionally, GRHL2 maintains barrier integrity and cellular polarity in epithelial tissues, with implications in cancer and epithelial mesenchymal transition, where it acts as a regulatory switch governing this process's promotion or inhibition. Understanding GRHL2's binding behavior to DNA holds scientific significance for deciphering transcriptional control mechanisms during development and in disease. Mapping DNA motifs recognized by GRHL2 can reveal insights into gene regulatory networks, aiding in understanding physiological and pathological processes. Identifying novel DNA binding sites or variations in known sites can deepen our understanding of GRHL2's specificity and plasticity in gene regulation, offering potential therapeutic avenues for diseases involving apparent cell proliferation and differentiation like cancer. Here, I start with raw DNA microarray data to investigate the protein's binding characteristics across diverse DNA sequences. Utilizing unsupervised machine learning techniques, such as clustering based on k-mer frequency within sequences, I hope to uncover motifs with strong affinity to GRHL2. This approach facilitates the identification of potent protein binding sites crucial for regulatory key target genes involved in neurulation and epithelial integrity, providing valuable insights into the protein's mediated gene regulation mechanisms. So now on to my process. So separately, before this project, I had already globally and quantile normalized my raw microarray data. So first, all I had to do was load and inspect the columns, look at the data type and the number of rows I was working with. For the second step, I looked at my k-mer counting and used a mer length of 8 because that was the length of the protein of interest's binding site, as I mentioned previously. For my third step, I employed PCA's cousin, truncated SVD, to reduce complexity from over 6,000 features just to 100 features. This helped make my processing speed a lot faster, but unfortunately also retained very little of the variance. But it was more of a proof of concept rather than a true investigational study. Lastly, I did this with k-mini batch means for clustering to increase speed, which just means that I didn't have to actually cluster based on or find centroids based on all of the points within a cluster. It would just randomly sample and use a smaller batch of points. This was necessary because, as you can see, the length of my data frame was close to 700,000, and because of that, it just took too long to try different clustering without randomly sampling and using k-mini batch means. To be thorough, I looked at silhouette scores of different numbers of clusters. On this graph, you can see the different parameters I ran this with. So at this point, I had a DNA motif length of 8. Variance retained was 6%. As I mentioned, that was quite low with after the PCA. And then with my mini batch means, my batch size was 80 and my data subsample was 50,000. So I played around with these numbers and generally found that 2 clusters was reliably the best based on silhouette scores. Because 2 clusters doesn't really make sense to continue with my analysis, I just kept all 9 clusters and looked at different binding intensities for each of the clusters. So you can see that here in the figure to the right. So looking at group 9 from the previous slide, I wanted to just see which sequences had repeated motifs. So here's just a count with the binding intensities with the sequences with the highest binding intensities. So as noted in the introduction, AACCGGTT is the sequence with the perfect binding site matching the literature for the protein of interest. This analysis yielded a number of somewhat similar sequences, as you can see to the right. However, it seemed to be more likely to me that they have been selected alphabetically rather than due to preferential binding. That said, if this error is ruled out, this would be an extremely useful result demonstrating that there are likely multiple binding sequences for GRHL2, and that a sequence of adenines is the most important component for this protein to bind. Regardless, this exercise is a proof of concept that it is fairly straightforward to group sequences based on base similarity and correlate these groups with experimental binding intensity. Further tweaks are needed still for the method along with domain knowledge of GRHL2 domain binding specificity, and for that I suggest bringing in bioinformatics experts with more expertise in genetics than I have. So there are a number of things I could have done to improve my model. First, I would probably reevaluate my clustering and whether it really makes sense to do it in the way that I did. I started out by looking at similarities between DNA sequences and clustering based on that, but perhaps it is better to change my method and look at how they form structures rather than just the sequences alone. Other things I could do are just reducing the complexity of the problem to decrease computation time, or keeping the complexity and running my program overnight or waiting hours and hours, or increasing my computational ability just to include more of the data in the model for better training. And that way I can open up my parameters to including more groups or changing the size of batches. As I mentioned before, consulting with industry experts would be a very good thing to do. And then also considering other applications of protein DNA microarray binding with this data, as it could answer multiple questions, not just the one that I asked about with this particular protein. Thank you so much for listening, and if you'd like to check any of my references,